Welcome to our Bone Lab podcast, and here we're going to go over all the visual identifications of everything related to bone. Now, in this lower magnification, we're looking at a cross section through a long bone, probably the diaphysis of that bone, and we can see that we've got compact bone around the periphery. We have some skeletal muscle attached here, and on the inside of our marrow cavity, we have some spongy bone. Now we're going to continue to zoom in on various regions that you'll see insetted in our little box in this location as we get higher and higher magnification. Here in the inset picture, we can see the periosteum, our two layers. We've got our fibrous outer periosteum, our intercellular periosteum. A lot of the eosinophilic, this red material that we see, this is going to be our true bone, secondary bone. Remember, this is going to take the place of primary bone, which is basically calcified cartilage, and this basophilic blue material is going to be that calcified cartilage or primary bone. You can see those different colors. Here we have some more, and then here we have some in this location. Here we have cavities where we have blood vessels and other cells. The flattened cells that line these are going to be bone lining cells, and we're not going to pay a lot of attention to those. We're going to be looking at these much taller cells that we see lining these cavities, and these are our osteoblasts that are actively laying down bone matrix. Now, if you notice just beneath the osteoblast, there's always going to be a clear area. This is going to be the uncalcified organic components of the bone matrix, and we simply call that osteoid. Now, nuclei that are completely surrounded by the bone matrix, these are our osteocytes. And then we have other cells that we can see associated with bone that are multinucleated and extremely large. These are going to be our osteoclasts. These are our bone resorptive cells, and we're going to look at these in a little bit higher magnification as we go through. Here's our inset with the periosteum, the outer fibrous, intercellular, our blue osteoid, and then our bone, our secondary bone. And here we can see a small one, multinucleated one here, large multinucleated osteoclasts. We've got our osteocytes surrounded by bone matrix. And we really don't see a good osteoblast that I'd like to point out in this one. We do have some osteoprogenitor cells out here, and we're not going to visually identify those. Once again, we're looking at a cavity where we have our osteoblast adjacent to the bone. Nice white area of osteoid. Mostly we're looking at bone, but we do have some of that calcified cartilage that we can see in this location, our really tall osteoblasts, and then our osteocytes completely surrounded by bone matrix. Now, in this particular illustration, we're looking at a neonate. We're looking at a suture line, a point to where we have the fontanelles or the bones of the cranium that are going to be sealing together slowly over time. I think you can probably recognize in this image that this is not really looking like bone. We have these large cells, lots of lacunar white spaces. This is going to be the hyaline cartilage of our nasal septum, I believe that's what this is, or the nasal bridge. And then we've got our bone here in this location. We've got some of our osteoblasts, a lot of progenitor cells that are going to be in this neighborhood as these two compartments are growing together. So again, looking at a bit higher magnification, we got a nice osteoblastic cell here adjacent to the bone that's being laid down. Some bone lining cells here, more osteoblast, and an osteocyte that's completely surrounded by this eosinophilic, this red bone matrix. Now we're looking at other areas where we're looking at bone, and now we're getting to the point that we see these thin processes of bone. These will be either spicules or trabeculae of bone as they get larger. We've got a lot of cells running around in here, but the ones that you can immediately draw your attention to are these large cells, multiple nuclei, and they're almost lined up in a row. Here we see one, another, here's one, and possibly one over here. These are our osteoclasts, the bone resorptive cells. Then we've got our osteocytes surrounded by the bone. Here we have our osteoblasts, taller cells laying down that bone matrix. Now we're getting to our epiphysis, and actually the epiphysis is going to be in this location. As we move toward the diaphysis, we're going to get into the metaphysis, or the middle portion, and this is where we have our epiphyseal growth plate. Here we have our secondary ossification center in the bone marrow. Here we have our secondary ossification center in the head of the epiphyses. And then right now, this is a fairly young bone because we've got hyaline cartilage on the surface of the epiphysis and here in our epiphyseal growth plate. So once again, as we're looking at our bone matrix, we see in the inset, we can already recognize that we've got these large multinucleated osteoclasts. We have our bone matrix. We even have the beginnings of an osteon forming here. You can see the concentric rings around the central canal. Here we have our bone lining cells and osteoblasts laying down bone. A lot of osteocytes here surrounded by our bone matrix. Higher magnification, once again, we've got our osteoclast, and I really like this osteoclast, multiple nuclei, but here you can see this shallow depression, almost like a bowl, present here in the bone. This is the area of bone resorption, and this is going to be our Hauschup's lacuna. Here you can see another depression angled out here, osteoclast. So Hauschup's lacuna, osteoclast, osteocytes present here. Possibly osteoblast, but being isolated like that, I would never ask you that on a quiz. So here we have another cross section of bone, and you can really see this is more mature bone. We're looking at a lot of blood vessels, and you can see all the red in here. These are the blood cells. So very thick, compact bone on the periphery, spongy bone lining the marrow cavity in here. So looking at a higher magnification, again, we're looking in the region of our compact bone. We can see our central or Haversian canal systems cutting cross section. But then we also see these perpendicular extensions extending between different Haversian canals. This is a Volkmann's canal running perpendicular. And then here we have basically the cementing lines of our circumferential lamellae, those plates that go around the edge of the bone before we get into our osteons. Again, looking at a section through bone, compact bone on the outside, large marrow cavity, and these bones can have differing sizes of marrow cavities, different thicknesses of the compact bone, depending upon what the tissue, what the pressures have been subjected to in that particular organism. And again, these are remodeled throughout the life of the organism. So once again, we're looking at the outside of the bone. The periosteum would be here. We would have our circumferential lamellae. We have our Haversian canal systems. And then we have our Volkmann's canals, which you see in longitudinal section. That's how you're going to know they're Volkmann's. Here we have a nice osteon, and you can see the Haversian canal cutting cross section, almost just the size of that red dot. When we see these in longitudinal section, then we've got a Volkmann's canal. Here you can see our inner circumferential lamellae, the plates. And then can you recognize this tissue here? A lot of that's going to be white fat, unilocular adipose tissue present in that marrow cavity. Now we're looking at the osteons that we have present here. And so we can see 3 Haversian canals that are present in the center of these osteons. And as we find the outermost stretches of these, that's where we can find the cementing line, which is going to be in this location for this particular osteon. Here we can see a cementing line for this osteon. Just a hint of a Haversian system here, and possibly cementing line in this location. So this is our osteon and the lamellae, the circumferential lamellae of our osteon. But then again, we have some lamellae that are present not associated with any of these intact systems, and these are going to be our interstitial lamellae in between our osteons. Once again, a number of Haversian systems, circumferential lamellae. And do you remember which is going to be the youngest and the oldest when we look at a particular osteon? The oldest is going to be the most peripheral, and the youngest is going to be the one adjacent to the Haversian system. So essentially, the Haversian system started with an extremely wide Haversian canal that, with concentric rings forming over time, it just gets more and more narrow as we go. You can see this one is pretty wide, a little bit more narrow. Here is a very, very small diameter Haversian system. Now, this is cut a little bit differently. We're almost getting into our inner circumferential lamellae. You can see how these are streaming along. Here we have some osteons, very sort of abstract in this. You don't see the nice circumferential lamellae in this one. This is going to be a phase contrast image because it almost looks 3-dimensional. But we're still looking at bone, a lot of osteocytes present in this particular micrograph. Now, often when you look at bone, it's going to be demineralized bone. And this particular image is going to be just that. They have demineralized, tried to remove the calcium phosphate matrix. And so they've stained this with a silver type stain to try to stain the plasma membranes of cells. And that's what's going to give it this brownish type color. And with this, we can really begin to see the Haversian systems, but what we're looking at is not the bone themselves, but the darker color are going to be the nuclei of the cells. And in this case, most of what we see are going to be the osteocytes, and that's the darkest material. And the lighter brown material that we see are always going to be those canalicular processes extending from one osteocyte to the next. Here we have a nice higher magnification view, osteocyte and all of the canalicular processes extending away from this cell. Here we have processes extending to and from these two. Here we have processes that are going to be extending forward to the next cell, this one extending forward. And so all of these are the canaliculi, and these are the nuclei and the cell bodies of our osteocytes.